This is the first prep, maybe the only, for lecture 26. I've cut lecture 26 into two shorter ones, and my idea is that we'll get caught up and we'll get back on schedule because there's a few lectures that are kind of slopping over into the next topic. Okay, so we have talked about in previous lectures, we've talked about mesoderm patterning, and we've talked about somite, the formation, the timing of it, and then the patterning of the somites. And now we're going to look at neural tube formation, and in the next lecture we'll talk a little bit about neural crest here, but we'll spend more time on it in the next lecture, lecture 27. And there's a set of learning outcomes that will be in your notes. So the neural tube is going to give rise to the central nervous system, and that means the brain and the spinal cord, but not the peripheral nervous system. The peripheral nervous system is neurons that emanate from either the brain or the spinal cord to innervate your muscles and to innervate sense organs and that sort of thing. So CNS is just the brain and spinal cord, and it is all formed from a very simple tube, but the tube at the anterior end is elaborated greatly, and we'll spend a little bit more time talking about that elaboration, just a bit, into a much larger, so there's, what's the right, it's not expansion, but it, I forget the name, it'll come to me, old brain, but it basically opens up, dilation. And it creates different regions. We won't really talk about the folds and that sort of thing. We'll talk about sort of very, very basic divisions of the early brain. But just note that there's a series of ventricles that are shown in blue here, and these are spaces in which cerebrospinal fluid is present and circulates. There is a little bit in the neural tube as well, but most of it is in this region, and it's part of sort of protection of the brain, but also nutrition. It's delivering nutrients and so on. Okay, so when we talk about neurulation, we're actually talking specifically about the formation of the neural tube. And so neurulation is, you start with, the ectoderm. The ectoderm can form epidermis or neural plate and neural crest, which we'll talk about. And what happens is the first part is that the neural plate, cell movement and cell shape changes create a much thicker area where the neural tube is going to form. The cells change shape too. They're columnar rather than shorter and wider. And then there's a series of cell shape changes kind of around different regions of the developing neural tube that help to shape the actual tube. The key events, though, are that we're going to look at the molecules, just a small number of them, that are differentially expressed in the epidermis versus the presumptive neural tube. And then there is a population of cells on the boundary between the epidermal and neural tube cells that are going to form the neural crest. And these actually kind of delaminate away from, so they don't maintain connections to each other. They become individual populations of cells, and they basically migrate down and around the somites, which we'll talk about more in the next lecture, and they seed lots of developing organs and tissues. So we'll talk about, they're a little bit about, they contribute to heart innervation and gut and some other tissues, which we'll talk about more. And the cells of one end of this tube and the other are going to fuse with each other to form the tube, and then the presumptive epidermal cells are going to meet each other and fuse with each other, and that kind of leaves out the neural crest cells, which then, as I said, they are kind of excluded from either of these two tissues, and then they start delaminating and detaching from the other cells and then moving away. So basically, this is neurectoderm tissue, and sorry, this is neurectoderm tissue, and this is sort of epidermal tissue on the side here. And so, as I said, we get a thickened neural plate. So there's some change in cell shape, and usually, if you look at the width of these tissues and these tissues, the ones that are going to form the neural tube are a lot thicker. And then convergent extension happens to sort of elongate this region so that it forms a larger, larger area, because it has to reach around and make a tube. And there is remodeling of adherens junctions. So this allows the cells to form a nice, neat sort of set of raised structures here. And we'll be talking, I'm just getting distracted because I'm thinking about what I will be saying later about the adherens junctions. We will talk about those just briefly later on. And so the first thing that you see is that there's a bit of a furrow. This is the first place where the cells are kind of changing shapes, and they're getting narrower at one end, and they remain the same width at the other end. And so you get this kind of constriction. And this happens through the changes in the actin cytoskeleton. And you might remember the thing I was distracted by was just a reminder to myself to mention and remind you, because we've talked about this before, that junctions like adherens junctions, tight junctions, and so on between two cells also involve connections to the cytoskeleton. So that a change that happens in this cell, if this cell sort of changes shape, that information, the tension that causes, there's a little bit of a pull maybe on the adherens junction, or a push, it depends on what exactly is taking place. But that information can be communicated just by pressure and movement to the cell next door. And so that's an interesting part of this whole process. The other thing, that I was thinking about is to just to mention that when I'm talking about neurulation, instead of having a whole wide variety of different lectures on this topic, because there's lots of interesting stuff, I've selected just bits and pieces from this part of chapter 5. And chapter 7 and chapter 11. So we have the intro and then sort of extra details on certain aspects. And I've tried to select enough to make a kind of complete story without kind of overwhelming you. I mean, we could spend really, literally the whole semester on just neural development and pathfinding and, all the different kinds of neurons and that sort of thing. I'm trying to keep it pretty basic. So we get this shape change that generates this neural groove, and then the neural folds are these regions here, and they are based on continued growth of the epidermal cells. This shape change that occurs right in the neural groove that alters the shape of the structure, and then some outgrowth and extension of the neural plate itself. And so this leads to this kind of uprising. It looks like little waves on a surface of the neural folds, the lifting of the edges. And that's pressure probably from both sides that leads to that. And then as the neural folds continue, there is some growth and upward push from the epidermal tissues. And then as I've already mentioned, the cells are approaching each other, and what happens is the edge of the epidermal cells find each other and fuse. The edges of the neural tube, the presumptive neural tube, they find each other and fuse, and that excludes the neural crest cells, which are left between these two completed structures, and then you get migration away. And a variety of routes. So different of these cells are going to wind up in different regions of the body. Now this is happening very early in the mouse, so day 7 to 9 after fertilization, but it's weeks 3 to 4 in the human. After implantation, as you will remember, the human development takes much, much longer than the mouse development, even though in the earliest stages they're very, very similar in terms of timing. So if we were to look at the chick embryo, this is kind of what the neural furrow looks like, and you can see the neural plate here. The cells are differently shaped, so they're taller and narrower, and these are the sort of wider and flatter epidermal cells. This is what the grooves actually look like. You can see sort of the epidermal cells on the side here, and how much thicker these neural tube cells are. And the neural crest is going to be this little bit here and this little bit here. And then the folds actually come in contact with each other, and then you can see the formation of the tube, and you probably, it's not very clear at all, but you can probably see sort of the connection between the epidermal layers, and then neural crest is going to be in between here, but it's not very clear, not very easy to see. So the apical restriction begins in the place where the the neural furrow first forms, and this is called the medial hinge point. Medial, it means middle because it's right in the center, the midline of the body. And these are apical constrictions, so cells have apical and basal polarity. And what happens is the networks of actin fibers across the apical side, they contract, they pull the apical part narrower while leaving the basal part the same. And when that happens, in order for the cells to maintain their contacts with each other, I'm not very good at drawing this, but the idea is that when you maintain these contacts but narrow these surfaces, you're going to necessarily form a little bit of a groove, right? And that's what this is showing. Now, 1 or 2 cells isn't going to do it. See how narrow the cells are across here, and the actual difference between the apical and the basal sides is really small. So this is greatly exaggerated. It's just a little bit narrower. But when it happens with lots of cells, you can start to see the effect it's having. And then there's a second. So most of these cells are going to eventually slightly narrow, but the second event is sort of at what we would call the hinge points. These are dorsolateral because they're on the dorsal side, and they're more on this structure. They're lateral because they're further away from the midline. They're more to the side, basically. And so these hinge points help the cells kind of bend around and reach each other. I'm going to tell you something else. I'm trying to remember what it was. But we'll just leave it, I think, at that point. Just the idea that it requires the cells to change shape to make the entire structure alter its shape. And it starts at particular locations. And the reason why this works the way it does is because these cells have maintained some connections with each other. And some of these shape changes can be communicated to nearby cells so that you get this nice rounded tube structure. Okay, yes. And so, and then eventually what's going to happen, as you know, as we've already talked about, is those neural tube cells will fuse with each other, and the ectoderm cells will fuse with each other, and there's a little bit of cells in between that's going to form those neural crests. And this is just a reminder that this is happening after the Henson's node is passing through. So Henson's node is here. The neural plate starts to form, and as the Henson's node is moving along, there's a certain time lag before the hinge point, the first one, the medial hinge point occurs, and then a little bit later you start to see the dorsolateral hinge points, and then much later on a complete closure of the tube. So this is just reminding you a little bit of the timing. We've already looked at this part, and this is just connecting what we've just talked about to what we covered in a previous lecture. And this is just a reminder. I've already kind of talked about this, and this is a better image because this shows you how the apical constriction basically is relatively small, but that it can be connected to communicated to nearby cells, right? So this kind of covers the things that I've already talked about. And it turns out that cell adhesion molecules are necessary. They are necessary between these cells so that when one or two cells starts constricting, the other ones get the message that we're constricting, so that you can start having all the cells properly participate in the formation of the tube. But then later, when we're starting to fuse the correct cells together, the epidermal and the neural cells, cell adhesion molecules are responsible for that process as well. So this is, I've color-coded this to hopefully make it clearer. So if you look at the whole region with the where the neural plate has formed, both the epidermis sort of and the presumptive neural tube, so the neural plate, all of this region, so basically all the ectoderm nearby, is going to express L-CAM. C-A-M means cell adhesion molecule, so they're called CAMs. And L is just that it was first discovered in liver tissue. But again, just like a lot of these molecules that are named after something that they were found to do when they were first discovered, it's been found that a lot of them are expressed in lots of different tissues and do lots of things. So this is the liver-specific cell adhesion molecule, but here it is in the presumptive neural plate. And it's basically helping all these cells stay together. And then as the neural folds start to develop, what happens is that the neural plate starts to express different ones, N-cadherin and N-CAM. So N stands for neural. So they have, they're expressing their own adhesion molecules, and the adjacent epidermis starts to express ECAD. E stands for epidermis, so it's first found in epidermal tissue. And these changes in adhesiveness, we know that these are all homotypic interactions, which means ECADs can bind to ECADs, but they cannot bind to NCADs or NCAMs or any of that kind of thing. And the same with the N-cadherins. They can only bind to other N-cadherins and not the other type. So the idea is that as these cells are moving together, and there is a region in between, perhaps it's expressing either none of them or perhaps both of them, I'm not sure. But the epidermal cells that are expressing the same one, they have this homotypic interaction, and so they will be able to bind to each other. The N-cadherin expressing ones will be able to bind to each other, and that's how the tube gets closed and how the epidermis is sort of finished. And then there are still those remaining cells in the middle that are going to form the neural crest. And this is just a reminder, in case I forget to mention, that these are homotypic. So all the cadherins, and remember that cadherins are found in adherens junctions, and they are homotypic. That means they will only bind to others of their own type. Okay, and they also, I don't know why I said remember, because I don't think I've mentioned this before, but cadherins also require calcium for binding. And there are a number of processes in development where calcium is one of the signaling molecules, and if the signaling leads to different cell shapes or different cell movements and so on, it's possible that the effect on cadherins is part of that process. So that's just a little extra piece of information. We're not going to do anything particular with it. Um, so this is one of the experiments that basically showed that probably these cadherins have roles in the proper folding up and fusion of the epidermis, right? So here's our normal development. We've got our NCADs expressed in the neural plate. The presumptive epidermis has ECAD being expressed, and then normally the correct cell types fuse with each other to form your overlying epidermis and your neural tube. Okay, but if you actually do an experiment where you inject into the presumptive neural tissue ECadherin, so that now it's expressing both NCAD and ECAD, you don't get proper closure of the neural tube or of the epidermis. And so it's necessary that this tissue be expressing a different cadherin than this in order for these two parts to meet and bind to each other. And it's important that just one cadherin, the same one, be expressed in all of these cells in order to close this tube. So both the closure of the tube and then the fusion of the overlying epidermis requires that these two cell types are expressing different cadherins, right? Yeah, so that's just one of the kinds of experiments they did that shows that the type of cadherin present really matters to this whole process. We also know that the notochord has a role, and the role isn't necessarily in inducing the neural tube to form, although it probably does have signals to that effect. But what we're going to be talking about here is just the dorsal versus ventral patterning of the neural tube. Under normal circumstances, the notochord is actually like right here. It's not miles away the way this picture shows. It's actually much closer. But you have the presumptive neural tissue from here all the way around to here. And when this tube forms, because as the tube is forming, it's very close to the notochord on the ventral side, signals from the notochord initiate gene expression in this region, which is referred to as the floor plate because it's kind of at the bottom of the neural tube. And then you have ventral specific fates, dorsal specific fates, and then what is called the roof plate. When this fuses, it's going to be up at the top. So this is going to be, your roof plate, and this is going to be your floor plate. And then if you look at the neural tube all the way down the spine, because this is a spinal one, there's a whole batch of different kinds of neurons. I don't think, this isn't a neurology course, so you don't need to learn any of the names. I'm going to show you a picture in a minute that shows lots of different names. But the idea is just that sensory neurons are extruded from the dorsal part of the neural tube, so they're receiving information from receptors, like taste and touch and heat and cold and sensation of movement and all that kind of thing. So your sensory perceptions are coming from outside the body, and then you detect them, and then that information is sent to the spinal cord through those neurons. And then motor control is innervating muscles from the neural tube, the spinal cord, in the ventral side. So there are different kinds of neurons on the ventral versus the dorsal of the neural tube. Okay. And so this doesn't tell us anything about what the notochord is doing, but this is a piece of experimental evidence that shows us a little bit about how that works. So if you were to, during embryonic development, this is almost certainly in the chick embryo, if you were to take the notochord from another embryo and transplant it while the tube is closing, you can transplant it very close to, one side. And what happens is one side of it, the one that doesn't have the extra notochord, that basically develops normally. So you have your floor plate, you've got your ventral, your dorsal, and then your roof plate. But where you have placed the extra secondary notochord, it's induced a second floor plate, so that's the floor plate there. And then there's a whole area here that's induced more ventral fate, and then some dorsal and so on. So we know that signals from this notochord have basically specified both the floor plate and the ventral fates to either side of it, right? So we have more ventral tissue than normal, and it coincides with exactly where you've placed that secondary notochord, right? So that shows us that the notochord has a role in this process. So what are the signals? It turns out that our friend Sonic Hedgehog, which we've been talking about before, Sonic Hedgehog is one of the signals, and it's probably the most important. And when we talk about dorsal ventral patterning of the neural tube, we are only, is that true? Yes, I think I cut out some of the other material that in the lectures that are coming up. So we will only probably be talking about Sonic Hedgehog. So basically, there is signaling to basically the central part. This is where the ventral the neural furrow, neural groove is going to form. And Sonic Hedgehog starts being expressed in this region. And Sonic Hedgehog is a diffusible, fairly short range signal. So the signal is going to emanate outward. So there's going to be tons of Sonic Hedgehog at the source, and then less and less and less as we move away. Now, the epidermis is expressing BMP, and the BMP expression is strongest in the epidermis, but it also is being expressed in towards the neural the neural folds at this point. Now, Sonic Hedgehog actually has sort of restrictive signals, so it's going to try to prevent too much expansion of the BMP signaling in this direction. So that's the overall. So there's antagonistic reaction between the expression of Sonic Hedgehog and the BMPs. The strongest BMP expression in the neural tube is going to be up at the top, the dorsal most part, and the least as you move laterally and down towards the ventral side. The most Sonic Hedgehog is going to be in the ventralmost region with less and less and less until there's essentially none in the dorsal side. No, I didn't eliminate. So there will be a couple of genes. So basically, the roof plate is the source of BMP signals, and these BMP signals are specific for dorsal differentiation. And so these are the three transcription factors that are expressed in the dorsal. Oops, the dorsal side. It's almost the same color. On the ventral side, we have Sonic Hedgehog. We've got BMP4 is reduced, but for the ventral, we don't have MSX, PAX3, or PAX7 that are dorsal specific, but we do have PAX6. So PAX6 is probably the only one you need to remember here. And what this leads to, I have a picture coming up. So I don't want you to worry about all these they're actually quite simple to remember because they're just numbered from the roof plate around, and then when you get to ventral, they're numbered down to the floor plate. But just don't worry about them, and you can just remember BMP here, and then Sonic Hedgehog here. So the idea is just to remember that as you move around the neural tube, there's all different kinds of neurons. The neurons that are innervating from sense organs on the dorsal side, different ones, different kinds, including some that actually connect the motor neurons and the sensory neurons. And then innervation of different muscles and different regions from the ventral side. So there's in different locations around from dorsal to ventral, you're going to have different kinds of neurons, and they will be specified by the amount of BMP. Lots of BMP at the top, less and less as you move around, and then, pretty much zero when you get to the midline, and then a little bit of sonic hedgehog, and then more and more and more as you make your way around to the ventral side. And each of these has different fates depending on the level of that signaling. So that's the main point here. It's not about trying to memorize everything, and you don't have to worry about, these gene names. So this is a picture from a previous lecture where we talked about how the notochord and then both the ventral and the dorsal neural tube, as well as the epidermis and then the lateral mesoderm, are sources of signals that tell the somites what they're supposed to be forming. So the sclerodome, which is on the sort of ventral kind of medial side, is receiving these sonic hedgehog signals too, and that's helping it promote scleroderm fate, which is going to form the vertebrae. And then the BMP4 signals here are telling the dermamyotome that you're going to form muscles, and it leads to enhancement of muscle-specific genes and so on. So that's a somite patterning. And just to point out that the neural tube, while it's receiving the signals, this signal from the notochord, and while it's been giving signals to help pattern the somites, it's also being patterned itself by these same signals, right? So it's happening at the same time. It's happening via the same kinds of signalings from the same sources. Okay, so that's just to show, we've talked about these in two separate lectures, but they are similar and connected. And then this is just a summary slide, which you can easily go through. I didn't talk very much about Hox genes in the neural tube or the hindbrain. I will perhaps talk about it a little bit later, maybe very briefly in the next lecture. And that's it. So in the next lecture, we're going to finish neural development, and we'll talk about neural crest, and you will find out a little bit about what causes this color pattern in this cat.